I'm delighted to introduce Professor Stephen on to this event today, who will be discussing the fascinating and complex subject of microbiology. A little bit about Stephen. He undertook a PhD in public health, England's central reference laboratory in London, developing methods for the identification of certain important foodborne, human and veterinary pathogens. From there, he moved to the Danish Institute of Food and Veterinary Research in Copenhagen, where he established his own research area before relocating to New Zealand in 2005 to become leader of food program at ESR. Stephen was appointed associate professor at Lincoln University in 2015 and to full professor in 2022. And throughout the time, his research has spanned phenotypic and molecular diagnostics, gastrointestinal disease, comparative genomics, and microbiological aspects of wine and beer production. It's good to see that quick list. Some of the things, the roles that Stephen plays inside of the university. He is the chairman of the subcommittee on the taxonomy of Campylobacter and related bacteria. And presently serves on the executive board of the International Committee on Systematics of Prokaryotes. And he is a member of the science leadership team of the New Zealand Food Safety and Research Centre. Really welcome Stephen here today. Based on an introduction, look forward to hearing from him in this presentation here today. Welcome, Stephen.
Very much, and hello everybody. It's so nice to see you here. In preparation for this talk, I looked up a definition of research on the internet, lots of different definitions. And research really focuses on the discovery and creation of new knowledge, which is really about asking questions, isn't it? You ask a question of your academic subject, you get some sort of answer back, and based on that answer, you ask another question and you go forward. And this is the way that science tends to evolve. I mused over that definition. I thought, you know what? Your entire career, Stephen, has been based on asking one question. I hope that's not too disappointing. One question. The question is, what's this? And I'm not in the slightest bit disturbed by this, because this is quite an important question to ask. To illustrate this, I have a demonstration. You can see these two glasses contain some sort of a pale liquid. Could be anything. But they look similar enough to be, could be wine, right? Could it be? Could be an RTD containing pineapple. Could it be? I'll tell you what they are. One of them contains Sauvignon Blanc, the other contains domestic pledge. Now, if I drunk one of those, it would probably suit me quite well for this particular talk, you know, start calming down the nerves, start making me a little bit happy. Drinking the other one is like to take out most of my gastrointestinal system laying on the floor, poisoned, and probably going to die within about an hour. One of those outcomes is better than the other. Although by the time I get to the end of this presentation, I think, I wish it drank the other glass. But you know, what's this? At the macro level, this is reasonably straightforward to distinguish, right? So just pick up these figures, knowing what they are. Give a bit of the smell. And that's not the one to drown my nose. It's just a little bit closer. Okay, what's this? That's at the macro level. We're talking about bacteria, and things are just a little bit different. The clue is in the word microbiology, microbes, right? So smaller than the naked eye, you can't see them. These are electrical aggregates of two quite different bacteria, very closely related, right? One of these organisms is found in association with most plants. The other has been associated with cases of septicemia and diarrhea, right? So if you're undertaking some sort of a diagnosis of a particular patient, finding out which one is which, whether or not it's something you just swallowed by chance, or if it's actually the organism that is making you ill, these are where the important questions sort of lie in terms of diagnosis, and it goes right across the whole spectrum. Well, these organisms are closely associated with an organism called Campylobacter, and I must say, in my research career, I've done quite a lot of stuff on Campylobacter, and this is why. It is the most commonly recovered cause of bacterial gastroenteritis in the world. This is the current New Zealand status, as you can see, and it's about 3 to 4 times higher than anything else that is recovered in New Zealand, and it tends to be pretty much the same all over the world. But I would draw your attention to the fact that there are some other beasts down here as well. So there's a whole bunch of other things that could cause foodborne disease. I'm going to pull on this particular estimate of gastrointestinal disease across the world. 2 billion cases of gastroenteritis due to 22 defined hazards, bacterial and microbiological hazards in this particular paper. There's more than that, right? In actual fact, some of the estimates go up to about 200. But if you take some other work that's been done on undiagnosed pathogens and what attribution they may play, then you can add in another 0.4 billion cases, right? It is worth bearing in mind that some of these metrics include the fact, and we all know this, we don't always go to the doctor when we're ill. So there's a British study that has found that every case that is reported to the public health system, 172 go unreported. So all in all, foodborne disease is a major issue. It has, as I've suggested, major health implications, which actually has a knock-on effect in terms of the economy. Right? If you're really not productive, if you really might be costing the health service something and so on and so forth. And in terms of export products, there are most certainly some foods that cannot be exported if it contains a pathogen. You don't really want to be exported if it contains a pathogen. Stop. We've covered the fact that it's usually an underestimation. It's a huge burden of disease. And at the end of the day, a horrid picture coming up in case we're a bit squeamish. You know, we're not going to get things under control if we don't understand it, if we don't understand what they are or where they're coming from. Now, I mentioned that I've done quite a lot of work on Campylobacter and related organisms in the course of my career, still do. This is the current state of play of what Campylobacters kind of look like. Now, at some point in time, all of those organisms, all of those genera would have been called Campylobacters. These have developed quite a lot. We have 20 different genera and about 130, I think, individual species. They kind of fall into 2 major ecological groups, ones that are associated with hosts, you know, animal hosts, and humans, of course. And then we've got a whole bunch of other things that are basically associated with predominantly fire. And I've also annotated, according to what I can find in the literature, organisms that are found, you know, associated with foods and those that are identified as known or emerging pathogens. You get the idea about how important it is with all of these different species, how important it is to basically assign some sort of a label, to be able to say it's this that's causing disease and not this. And if you found the causal agent, that's fine. If it's not, then you go to something else. Now, in the course of my career, I've actually got to describe a whole range of these things. And again, the reason that we want clear taxonomic definitions to actually call something a species is to be able to say what it is in a particular site, to sort of attribute a level of is it doing anything or not. And so that's what you kind of expect from taxonomy. It needs to be clear so that everyone can basically speak the same language. This is how it's done, and it is reasonably straightforward, I think, as an overall process. The details are a bit more involved. You want to define the sort of phylogenetic or evolutionary position, and that effectively means that you can place it into a genus. Then you want to do some genome-to-genome comparisons among other members of that particular genus to say, okay, it's genetically distinct. And last but not least, to actually need some phenotypic characters, some way of a routine laboratory in the middle of nowhere to be able to recognise that particular organism because it might actually be in the picture. These are the baseline criteria. Unfortunately, not everyone plays by the same rules. So we saw this proposal in 2018. Proposal will take one of those groups of Campylobacter and related organisms, which I'll talk a little bit more about. And they said, well, you know, we think we can split this into 6 different genera because, you know, this I mean, it's not even true, actually, I'm right up against it. But according to a comparison of whole genome sequences for these particular bacteria, they said, well, you know, they form different clusters, and we're going to call these genera. And then they tried to argue that they had some phenotypic characters that would distinguish those genera. To be absolutely honest, I and a few other colleagues, notably Bill Miller at the USDA and Peter Van Dam at the University and the Cornelius and the ESR, actually, we looked at thinking it just looks shocking. One of the most important things about this is a lack of comparability with other members of the other genera that are related to this. How can you say that we've got a new genera here if you don't actually have something that you have a reference point with? Well, and in fact, what we have done with our revised, it's quite a lot bigger. We've used more genes, so some of the individual positions of this, some of the other genera sort of out. But right, in effect, what happens is that once you start comparing Archobacters with all of the other genera, it's actually the most tightly defined unit that actually is. Phenotypically as well, all of these organisms hang together on the basis of 2 criteria. Now, Archobacter is a particularly interesting case from an ecological perspective. It's actually the most diverse of all of the genera in the Campylobacter at the phylum level. You've got a range of different species that are associated with land-based animals and indeed humans, and these are the ones that have been associated with human sex, right? You've also got a whole range of organisms that are basically found in the environment and or associated with shellfish. It just so happens some of these human pathogens here are also associated with shellfish. They've been found in shellfish and they've been found to cause human disease. In terms of shellfish, this is one of the things I thought would be interesting. No. And from a commercial perspective, this is a reasonable export market worth about $2 billion into the New Zealand economy. And it's sold up pretty much, so you'd expect it to be so. So I just looked through it and found it. And we found a species called Archobacter chlorophorus from half the batches of mussels that we examined. Interestingly, completely separately, this species has also been reported completely independently in terms of New Zealand's infections. I thought that was quite interesting. Even more interesting in one of our strains, we had something that we have determined as a virulence plasmid. A plasmid is a self-replicating piece of DNA, can be passed from strain to strain, species to species, and the reason we're calling this a virulence plasmid is that our annotation for genes suggested that we're a whole bunch of genes associated with specific virulence properties, and there are actually quite a few of them. So right from the start, you think, well, actually, maybe it would be a good idea to be able to identify and screen strains found in shellfish using some sort of a method. Let me introduce you to elastic light scatter analysis. This was a tool developed at Purdue University by my collaborator, Paul Wilson. And in principle, what this is a laser beam that shines through a cultured bacterial colony. The laser passes through this platform here, and basically scatters, the laser goes through the colonies and scatters the photons, so you get some sort of characteristic picture. If if you capture the picture, then you can see, oh, it looks like this. That cartoon doesn't really tell you what it looks like. That picture does. So what you see here are images from the elastic light scattering analysis of 4 different Archibacter species, and hopefully you can see they're all actually quite different. Once you start getting that kind of separation, you can start building up databases to basically accommodate what you're looking for. Now, I know that's a horrible table, but if you're a student, please don't show stuff like this, because you really shouldn't. The only reason I'm showing this is the fact that we've got quite a few of our emerging pathogenic Archibacter species here. Basically, everything in here is associated with some sort of food. And what we want from an identification system is a level of confidence that your organism is going to confidently identify as the right thing rather than something that's the wrong thing. And in order for us to assess that table here, we're looking for this particular value, this confidence cross-validation value, but basically overnight, so we are confident we're getting that with these particular organisms. So this looks like quite a good process in order to screen your different species. So, well, that's good then. Why don't we use something for something else? So let me talk about another organism. This is the latest data for eusiniosis in New Zealand. And it's not coming down any time soon. There are 2 pathogens that are most commonly associated with eusiniosis, which is enterocolitica and pseudotuberculosis. We don't actually know why this increase is happening, but you can see it's definitively happening. And worryingly, it's associated with a reasonable number of hospitalisations. Among the problems that we have in detecting Yersinia in first is the fact that everything else grows pretty much at the same rate. There aren't any specific antimicrobial agents you can add to APOS to knock it out to improve its recovery. So what tends to be used is the fact that the organism quite likes to grow at low temperatures, at refrigeration temperatures. The problem with that is that you're then stuck with up to 21 days for incubating the sample to basically get it up to a level where you can actually recover it on the antibiotic. You're trying to track down an outbreak, that's absolutely hopeless. So one of the things we've applied this approach to Yersinia. What you can see here are images of Yersinia enterocolitica, Yersinia pseudotuberculosis, and all of these things. These are pictures, images of organisms that are basically found in porkies. This was porkies is quite widely regarded as a major source of Yersinia. So that was a natural matrix for us to try. And what the system does once you've got a database is that you can look at a petri dish when you grow these things up, and the scanner will assign some sort of an attribution to it. So in other words, you can look at the plate and tell that looks pretty much like a target organism. The fact that we're not using refrigeration temperatures to culture the organisms means that we can get the recovery time down 3 weeks to 39 hours, which is quite robust. In terms of proof of the pudding, a couple of the things that we found were 2 individual strains of Yersinia. These were not seeded strains that we put into the mix to test it. These were novel strains. And indeed, one of those, I think it's this one, is Yersinia intermedia, which is also a human pathogen, but not previously described in New Zealand. So I think that's quite good. Sorry, I should have worried about that. My third story in terms of pathogens and food concerns Vibrio vulnificus. So this is an organism that is found associated with seafood. It has a very high mortality rate and causes basically aggressive septicemia, as you can see here. This is from a cage report and a pumpkin pie. 15 years ago wasn't a thing. And 15 years ago, we were also detecting its sister organism, Vibrio parahemolyticus, at very, very low levels. Guess what? Climate change is happening. So now we're finding it. Now we're finding increasing episodes of parahemolyticus, and we're finding them. So The thing about, and you might be aware there have been some outbreaks described in Vibrio paracampyloricus over the last 2 to 3 years, and they're just on the rise. Vibrio paracampyloricus isn't particularly well studied, and so in collaboration with my colleagues at Plant Food Research, we said, let's do something together. Let's do a project. One of the things we wanted to do was establish here whole genome sequencing methodology. And so, look, you know, you want to do genome sequencing well, and you want to do it right. So, ideally, for the best result in doing things, you actually use 2 complementary approaches to do it. You basically use a machine that produces long strands of sequence, and you use another one that produces short sequences. And ideally, what we do is put all this stuff together and get an ideal read. Really expensive. I mean, right from the start, you've got to have 2 quite different types of machines, so it's going to cost you a lot of money. So what we wanted to do was to establish something comparatively inexpensive but high fidelity. And we are very fortunate in this university to have an expert geneticist, Dr. Chris Whare. So with me and a very clever PhD student, Whare Wānaka, as well as Chris, kind of put our heads together and thought, okay, is this method going to work? And that's actually the results from our first experiment. Might not look like a lot to you. The other thing about Vibrio that I should mention here is that unusually among the bacteria, it has 2 chromosomes. Most bacteria only have a single chromosome. Well, first run, we're getting sequencing to the level of confidence that we can actually have complete chromosomes assembled. Here's chromosome 1, here's chromosome 2. What's this little guy? That's a chromate. That's a megapathogen. Never this before discovered in Vibrio paracampyloricus. Once you've got whole genomes, you know what I'm saying about identification and you know, the need for genetic dissimilarity. But you can use genomes for a priori identification too. You take your genome sequence and you challenge it against reference genomes. So we received 38 strains as Vibrio paracampyloricus. And what we found is that there were a few of those that were misidentified, illustrating actually it's quite hard to identify things from the start. 5 of those were Vibrio paracampyloricus, which I've mentioned before. These 2, Campbellii, and something that looks very much like, but might not be related to Rotiferanus. These are actually fish pathogens, and I don't think anyone actually knows about these, but potentially that's a bit of a risk for our seafood industry. So right from the start, even at the highest level, we're getting some really interesting sort of results. This is a comparison of key genetic traits of our strains against all other reference strains that basically represent individual types in a particular database hosted by Oxford University. The technique is called multi-locus sequence typing, just individual differences in particular genes. The coloured ones denote where our strains actually fall and everything else in the database. Things to note, all of the strains that we have possess at least one virulence trait, known virulence trait. This one has 3 of them, all of the ones that are definitive, so to speak. We've got another one down here. You can see that just from a genomic perspective, our strains from New Zealand are actually quite diverse, right? The strains here we've got most of are assigned to a type that was originally found in Norway, that has also been found in cases of human disease. So we can conclude that while some strains might pose potentially a bigger risk to people than others, they all have the same potential for harm. And we're going much further with this in terms of genome mutation. I think one of the things I kind of want to end on in terms of the genome side of things. I mean, I have a history, I think, in my career where everyone jumps onto the latest bandwagon. And no one really pauses to think about, well, you know, how do we use this and what does this actually mean? But these are all nominally the same strain. The lower one is basically from the database. It's from GenBank, and this has been assembled with the gold standard, you know, 2 complementary sequencing technologies. And this is the way it looks. This particular section. These 2 have been assembled from our data. This is our stuff, our sequencing. But the differences are because we're using 2 different assembly programs. And people I don't think people think enough about this stuff. This is quite a common one called Fly. And what you see, and this is one called Tricycle, which is a bit more recent, and it's effectively a consensus build between each of 3 different assembly programs. And what you see here is that there's a much better agreement in terms of the gold standard with Tricycle than there is Fly, which sort of breaks some genes up and then puts in hypothetical genes somewhere else. The other thing that is notable is that when you use these different programs, you get different estimates in genome size. Now, you remember I was speaking and showed you that tree with all of these different groups. Some people are now trying to define species on the basis of, well, it's 0.1% sort of in or out, so it must be different or the same. And my argumentation for identification particularly is that it needs to be a consolidated whole. You don't just rely on a single metric, it's certainly not when it's that close. These are living organisms that we're talking about. It's biology, it's things are going on different, and 0.1% is quite clear. Have I done enough speaking about the bad? Let's talk about the good. Let's take another sip of wine while I show you that slide about the wine industry. Again, a very successful industry, $2 billion worth of export, and actually beer, while it's not as big an export market, it's actually estimated to be a $3 billion market in terms of both domestic and export. So it's really quite burgeoning. Little things that you can see here is that these different classifications of beers, you can see that there's been some variance over the years, right? And you can see it in the supermarket shelves. Now, if wine is going to continue its expansion, and if beer is going to continue its expansion, then they're going to want different products. Different markets have different needs and different tastes. So sometimes you actually need to try and develop a particular product and for a particular market. Now, fermentative microorganisms, predominantly yeast, but also bacteria, play a critical role in product development. Just think about this. Without yeast, it's just juice. That's all you have. I don't expect you to read all of the attributes here. During fermentation, microorganisms, particularly yeast, that's on the grape juice and anything else it can get its hands on, which includes the water used to make beer and sort of apple juice and cider. But a whole range of different chemicals are produced during that process by the individual yeast. And each individual yeast strain will produce a different amount of it. So this is effectively part of the ability to get a wine that is going to win you a gold medal, or something that you just, clean the tables with, that potentially is part of the trick here. And I think we've been privileged actually to work with, thanks to my colleague Ben Tian, who's links with Greystone Wines. Greystone, this is an interesting winery. They produce wines completely organically, and what's more, they produce their wines in 2 quite different settings, in basically indoors in a winery and outdoors in a vineyard. But they were curious about what's happening in terms of the microbiology here. So I just want to show you a few things about our studies there. And our first set of studies, we partnered up with AgResearch. So we could use their matrix-assisted laser desorption time of flight analysis. Basically, it's a mass scale. It's a process that breaks down the cells that you introduce it to, separates all of the fragments out on the basis of their maximum charge. And this is the sort of thing you get. one of the things that my then PhD student, now postdoc, John, who's in the audience, one of the first things we did was to clean up the way that everyone else made their samples, so that you could actually get better, cleaner profiles. One of the advantages about that is right from the start, you can actually see what you're dealing with. In terms of my presentation, the good, the bad, and the unknown. Well, here's the good. This is the organism, Saccharomyces cerevisiae, it is most commonly used to make wines, beers, so on and so forth. This is Brettanomyces, which is the spoilage yeast. So you want to be able to differentiate those organisms right from the start. But when we actually looked at the differences between, it's the same bunch of grapes, same batch of grapes, but you're fermenting them under different conditions. And just look at the difference in terms of your species distribution, really quite marked in actual fact between these two conditions. Let's just take this a little bit further. Let's think about metagenomics. I mean, so this analysis is based on yeast that you can grow, yeast that you can culture. The technique known as better barcoding or metagenomics takes a particular process called PCR that detects individual gene sequences of basically everything that's in your sample. You use different genes for bacteria, different genes for yeast. And in effect, what you do is amplify everything that's in there, separate them, sequence them, and identify them. So let's think about using that for wine. Okay, good. No. This is actually from this is a snapshot of the bacterial diversity. So this is from the same vintage as I've described before in terms of yeast, right? You don't need to see the details. The different colours, basically different genera, right? And what you can see here, once I get the is up, between the vineyard and the winery, again, just like in the cultured yeast, the populations are really quite different. What do you think about that? Now, that almost looks like it's a different planet, doesn't it? No. Oh, I don't know what's happened to the labels there. But there's just 3 years apart. And that's actually quite strict. The colour coding is exactly the same. We've managed to include climate data that we've dragged out from public repositories. And it turns out that using particular types of analytical techniques, sort of different types of displays and environmental fitting and this sort of thing. And this work has been done by our PhD student that goes out who's in the years, so you'll be familiar with this figure. When you include all of the climate data, basically it's temperature. Temperature seems to be the predominant driver for these particular differences. I think there are two things that strike me back. Some organisms are completely absent and completely present in either which I thought was quite extraordinary. The other thing I really want to point out is that I have to say that I don't think that scientists should just rely on black box technology. Oh, here's a computer program, let's use it. So I do like to try and relate what we actually have here to the actual data what we have. And I must say that I do feel that this is actually quite impressive. If there was any indication that climate change had the potential to change our wheat vineyards as well as our seafood sector, and probably everything else in between, I think I could do a better job of that. In terms of getting the unknown into the known, 2 minutes. Cool. Just want to go back to the Malditoff story and looking at yeasts and their attributes and what you could potentially use them for. So I've mentioned that you can use yeast strains for making beer and making wine. Well, it turns out that Malditoff, when analysed appropriately, does quite a good job of separating out these particular potentials. Now that's potentially interesting, isn't it? To take an unknown organism that you just isolate from anywhere. You can isolate them used here, everywhere. But to be able to potentially predict its functionality, whether or not we can use it to make a really good salve, a really good mop, a really good spell, isn't that potentially kind of interesting? So we've done some work in this area. So this is published, this particular one. And what we basically have here are groupings that are quite useful in terms of looking at what they sort of do. So these are quite tolerant, mostly whites, I think. These are definitely pre-blue predominantly. This one and this one are mostly reds. And these ones are predominantly whites. I think I've got those whites. So we're getting some sort of an idea that you can actually independently take a profile and assign it to. This could be useful for making whatever. We are looking a bit closer at what characterises these particular strains in these particular groups. But the other thing that we're quite excited about is the fact that we're also working with beer, and you know, that kind of thing. I think that hopefully speaks for itself, where you've got some major groupings with ale strains and wheat strains. We're going to hopefully take that forward with the use of a little glass. And that's hopefully this presentation has given you some sort of idea of where we can actually go to, taking an unknown strain and assigning, depending on the question you're asking, is it good, is it bad? Because it doesn't have to be unknown. I'm going to end on this particular slide to highlight the fact that no man is an island, and most scientific research these days are done in teams. And I've been privileged to work with a whole range of different individuals like Bill and Andy and James at USDA, Paul, Valeria, Purdue, Graham and Salisa at Plant and Food, Jeff and Stephen at Ag Research, and Preet at Lancashire, Pat Biggs at Massey University, Libby at who's director of the Food Safety Science and Research Centre, Peter at University again, and thanks actually to Don Maxwell at Gravestone Wines. Last but not least, my very talented collaborators here within the university, Chris and Ben, and last but not least, my brilliant students, because they are the future of research. Thank you very much.
Opportunity to put some questions. Good, bad, and unknown in this game. If you just state your name at the start.
Charles Murfield. Are you sure that was a climate signal and not just year-to-year variation you inherently get in agriculture that drives different quality vintages?
I think that's a really, you know what, to comprehensively answer that, you just want more data.
Yeah.
You know, more data. I mean, I love the fact that we've actually found differences here. I think one of the things that really strikes me is the fact that on some years we have things that are there that aren't there in the next year, and vice versa. And I must admit, of all the things that I saw, I thought that was absolutely astonishing. The results for modelling are only as good as we have two vintages. We've got publicly available climate data. Is that everything that climate is made up of? Well, no, it's not. So it's a mixture of observation and seeing what there is. If temperature is a major driver, that's what the modelling tells you. That's what looking at, and so hopefully people will get to see this. We just set this off for publication. I think there would be a lot more stuff that we would actually want to do over a longer period of time to say this is what's driving it. But I don't think I certainly don't think it's not the case. I certainly don't think it's not the case. And if it's not the whole story, then it's part of the story that I'm quite sure of. Good questions.
So for that saying about climate changes impacting on the variation of the microbes in the wine, does it has actually also have an impact on the taste of the wine from the same? Does it make differences?
watch this space. We have, I mean, again, you know, some of what I've described is really quite, what's the word for it, serendipity. So we're looking at, we've actually got some example wines made from the vineyard and the winery of one particular vintage. So we've got the story here in terms of their microbiology. And what we're going to do is marry that up with a taste test, as well as some biochemical profiling as well. So we hope to be able to answer that question soon-ish. I know a little bit, but not much.
Well, you're thinking I have questions around taxonomy, and you've given us a healthy dose of taxonomy. And one of those skills that's been lost from the past, the art of and science, you've demonstrated how much importance to food safety, and to I guess food in general, to wine and food production system. The question I have is there enough taxonomic skill in the right areas left to future-proof us from? It's quite a big question. Is there a big risk here that we're losing the taxonomic skills that you've spoken about? So, as you mentioned, I currently sit on the executive board for the ICSP, the International Committee for Systematic Pathogens. And my role is as an executive of the subcommittees. The subcommittees are groups of experts in particular. I mean, I've been chairman of the Campbell-Batts one for 5 years, and member for even longer. What seems to have happened over years, and I think it's quite a natural progression, is that expertise in particular groups is starting to diminish. It's being supplanted by people with expertise in the methods. So your genome sequences stuff. I and a number of my colleagues are a little bit concerned about that. And I don't think the situation is any different in what we're discussing at the moment, I think I'm actually the first secretary to have a cross subcommittee talk about what we could potentially do to sort of really integrate subcommittees and make sure actually that these skills are not lost, because they really are quite important. And it worries me when you see some of these proposals that I actually think are dangerous. But the rules of taxonomic nomenclature, I mean, it's a whole luxury skill. So I think the short answer is no. But I would like to hope, I'm an optimist, that we can do something to either arrest it or reinvigorate it. And I do think it's important. Okay. And thank you for mentioning that. One question.
This we can show the picture of the pictures, photographs, like the streets. Do you think this domain can need to be one way? How do you say that? How do you establish that?
So there are a couple of answers. It's very much horses for courses. There are some colony types that I know don't give you any discrimination at all. And one of the reasons for that is that the laser technology depends, it relies upon the beam going through something with depth. If you have a flat colony, that depth isn't there. So we already know that for some organisms it's a problem. I can't name them because basically the ones that I've worked with have got some sort of depth. So you get some level of discrimination, but I do know that there are others that are not the case there. So it's not going to work for everything. It seems to have worked for the things that I have played around with.
Supplementary on that, excuse me, would using different agars change the colony size and shape and everything else that would allow you to get better differentiation? So you can grow them on multiple agars?
Yes, we, particularly for the Archibacter one, although there are different challenges for we accounted for the boys in the Archibacter agar. We tried about 15 different variations But I mean, admittedly, it was slightly, part of that was getting enough nutrition from what is ecologically a very diverse range of bugs. So we had to do quite a lot of playing around with that. But the short answer is yes, and in some of the earlier papers done by other people on that technology, you'll actually see that they tried 5 or 6 different formulations of this, that, and the other from the colloid, or is the salmonella, staphylococcus, this kind of stuff. So yes.
Well, that brings us to the end of the time here, and I'll take the opportunity to thank Stephen for his outstanding presentation today, to bring to life particular issues around testing, really advanced techniques that would be put in place as not only in Stephen's own laboratory, but in collaboration with many others across the world. I think we've learned a lot about a sense of bad and good and unknown in this case, and particularly how these advanced technologies can be used to address issues of climate change, and that's certainly an emerging area that we can extremely benefit from. So thank you very much for your presentation today, Stephen, and it's been a total pleasure. When I was in the end of this building, it was the same.